Well, aloha, everybody. I am very happy to introduce Amanda Navine. She is going to present her master's thesis work on the project titled Investigating the Role of Colloquial Microbiome in Avian Malaria Susceptibility in Hawaiian Honeycreepers. Thank you, Amanda.
Thank you, Pat, for the introduction. Yes, I am very excited to share my research today on the gut microbiome and its role in avian malaria susceptibility in Hawaiian honeycreepers. So without further ado, we can get started. First, I want to orient you to the flow of my talk today. I'll introduce you to how the disease avian malaria is threatening the beautiful birds of Hawaii, then how the gut microbiome may be able to modulate malaria severity. I'll outline my research questions surrounding these topics and walk you through how I went about characterizing malaria infection and microbiomes for birds in Hawaii and what I discovered about avian microbiome diversity and bacterial abundance in relation to malaria. Finally, I'll expand upon some of the conservation implications of my research and the potential value of further investigation into antimalarial probiotics. So let's get started by introducing you to my study system, avian malaria in Hawaiian honeycreepers. Globalization and anthropogenic disturbances pose major threats to the conservation of biodiversity. One important example of a threat that is exacerbated by human expansion is the increased frequency and severity of emergent infectious wildlife diseases. Not only has transport and trade increased how often pathogens, parasites, and disease vectors are introduced to naive ecosystems, other anthropogenic stressors, such as deforestation, habitat fragmentation, and climate change, have increased the intensity of disease and lowered wildlife populations' ability to cope and recover. Altogether, human-introduced diseases have been the cause of numerous local extinction events in wildlife populations worldwide. This is especially true on islands, where species are particularly vulnerable, as they lack the immunological adaptations that their continental counterparts have evolved through historical exposure to these pathogens. Hawaii is known by many as a unique biodiversity hotspot, but what is less well known is that Hawaii is also the extinction capital of the world. Of all the species listed under the Endangered Species Act, almost a third are from Hawaii. Among the biggest threats contributing to the extinction crisis here are the various introduced species, such as non-native predators, habitat-modifying plants, and of course, diseases. From tree diseases such as rapid ohia death and koa wilt disease, which have taken out massive swaths of native forests, to toxoplasmosis, which is transmitted by cats, to our native monk seals, to various avian diseases, such as avian mange and pox, which cause large lesions on the feet and faces of native birds. While pox lesions like this one shown here look pretty nasty, pox is actually much less sinister than this last disease, avian malaria, which is where my interests lie. Many of the threats that I've mentioned have contributed to a dramatic decline in Hawaii forest bird populations, but the largest modern threat by far is avian malaria, caused by the blood parasite Plasmodium relictum. P. relictum reproduces in avian red blood cells, and when the parasite load becomes too great in infected cells, they burst, causing the bird to become anemic. Unlike continental species, Hawaiian birds have evolved in the absence of malaria for millions of years, and thus lack immunity to the disease. Upon the introduction of avian malaria's vector, the southern house mosquito Culex quincataceatus in 1926, we began to see highly elevated extinction rates among Hawaiian bird species, especially within Hawaiian honeycreepers. Hawaiian honeycreepers are in the finch family, subfamily Cardulinae, and similar to the famous Galapagos finches, are a prime example of adaptive radiation. It is hypothesized that a single finch species colonized the Hawaiian Islands 6 to 7 million years ago and evolved into more than 55 honeycreeper species, with a fantastic array of morphologies, behaviors, critical ecological functions, and cultural significance. Due to the isolated nature of the Hawaiian archipelago, the islands have a unique evolutionary history. Few animal taxa were able to establish here, and therefore birds fill many of the ecological niches that would otherwise be occupied by mammals or even insects in continental ecosystems. Honeycreeper species have diverse foraging strategies, from highly specialized snail eaters to generalists who eat a little bit of everything. But many honeycreepers are nectivorous, favoring the nectar of the bright red blossoms of our most abundant trees, Ohia lehua, as pictured in the top left. Honeycreepers serve as the predominant pollinators and seed dispersers in Hawaii forests, and as many as 60% of Hawaii's native trees are bird pollinated. The forests here are full of examples of coevolution between plants and honeycreepers, such as the iconic relationship between the dramatically curved bill of the iiwi and the curved flowers of the native Claremontia species, shown here in the lower right picture. As the most diverse terrestrial vertebrates in the islands, it's no surprise honeycreepers are prominent in native Hawaiian culture. Historically, these bright jewel-like birds were caught using a sticky sap applied to tree branches, and a couple of feathers were collected from them before they were released. The feathers were then woven together by the hundreds to craft intricate capes, lei, and headwear worn by ali'i, Hawaiian royalty. Honeycreepers are also the subject of countless mo'olelo, native stories, like the legend of La'iakawai, a beautiful chiefess who floated on the wings of the iiwi, resided in a house made of sacred feathers, and used apapane as her personal messengers. Unfortunately, Hawaiian honeycreepers are also one of the most endangered groups of birds in the world. Today, there are only 17 of the approximately 55 honeycreeper species left, almost all of which are listed as species of conservation concern by the IUCN. Most extant honeycreepers are restricted to high elevation refugia from disease, where mosquito dispersal is limited by cooler temperatures and malaria prevalence is low. But deforestation to make way for cattle ranching and introduced grazing species like sheep and goats have degraded the remaining high elevation forests extensively. And as climate change raises global temperatures, mosquitoes and malaria are expected to move up in elevation, reducing the amount of disease-free habitat even further. Without intervention, we will certainly see more honeycreeper extinctions in the coming decades. Successful honeycreeper conservation will require combating avian malaria from multiple angles, including reforesting high elevation habitat, controlling the distribution of its mosquito vector, and facilitating honeycreeper persistence in environments with increased malaria prevalence. And that's where the microbiome may come into play. There's building evidence to suggest the gut microbiome, abbreviated GMB, has significant influence over host health and fitness. Recent experiments with mice revealed a strong connection between gut microbiome composition and malaria parasite resistance and disease severity. Mice that are genetically similar but differ in their gut bacterial communities have been shown to differ significantly in their parasite susceptibility, burden, and mortality rates after exposure to malaria. There are a few proposed mechanisms explaining how gut microbiota can increase malaria resistance, centered around the ability of some microbes to prime host immune systems to fight infection. For example, some Escherichia coli species produce similar glycans to those expressed by malarial parasites, called alpha-gals. The presence of bacterial alpha-gals stimulates the host immune system to produce specific antibodies that can cross-react with malaria alpha-gals. These circulating antibodies then facilitate rapid identification of malaria parasites at the site of a mosquito bite and reduce transmission from skin into the bloodstream. Lactobacillus species also have documented abilities to modulate host immune responses, and as common vertebrate symbionts, they've been the focus of several anti-malarial studies. In a mouse model system, supplementation of yogurt spiked with Lactobacillus species significantly reduced the severity of malaria, potentially by increasing nitric oxide concentrations in the host's blood. Nitric oxide is produced by immune cells and at high concentrations has antimicrobial effects, which may suppress the growth of malaria parasites after infection. There are other microbes that have associations with malaria, though with less scientific support for their beneficial potential in hosts. One correlative study found the bacterial genera Bifidobacterium and Streptococcus were enriched in human populations that had lower risk of malaria infection. Pseudomonas, Pantoea, and Serratia bacteria have been found to block malaria parasite development in the midguts of mosquitoes, but no connection has been assessed between malaria severity and the presence of these bacteria in host gut microbiomes. However, one study did find a Pantoea species was in greater abundance in the mouths of bats infected with malaria. There are almost certainly others that have yet to be studied or even identified, especially in the microbiomes of less studied taxa such as birds. The discovery of these encouraging relationships between malaria and the microbiome in mammals illuminates a novel potential approach to the conservation of wildlife species sensitive to malaria, namely the development of anti-malarial probiotics. However, while the link between the gut microbiome and malaria immunity has been established for mice in a laboratory setting, little is known about the potential connection in other species or in natural environments. It is unclear if probiotics could be used for avian malaria control in Hawaii, but the recent expansion of one honeycreeper species begs for further exploration. There are 2 species of honeycreepers that are listed as species of least concern by the IUCN. Chief among them is the Hawaiʻi amakihi, scientific name Chlorodropanus virens. Amakihi are found throughout their historic range, including some areas of high malaria prevalence, and show variation in malaria mortality rates across an elevational gradient. Low elevation population mortality estimates are as low as 3%, but high elevation populations display as high as 68% fatality. This suggests that the increased disease pressure at low elevations has selected for the evolution of some immune strategies in these populations. Amakihi that survive malaria are never able to completely clear malaria parasites from their bloodstream and maintain chronic low-level infections. Contrary to what you might expect, one study has found that individuals with these chronic low-level infections actually have higher nesting success than uninfected malaria-naive individuals. It has been hypothesized that this is attributable to heritable malaria tolerance genes. However, there's an alternative hypothesis that has yet to be explored: the possibility that the gut microbiome of these birds can confer malaria immunity. There are numerous introduced bird species to Hawaii with historical exposure to avian malaria that do not suffer malaria-induced mortality, but serve as reservoirs for disease. One such species is the warbling white-eye, Zosterops japonicus, which was introduced in the 1920s and is now ubiquitous throughout Hawaii forests. Exposure to malaria in their historic range over evolutionary time likely favored the adaptation of a suite of immunological strategies, which allows white-eyes to clear parasites from their bloodstream, potentially including harboring a microbiome that can confer malaria immunity. Once introduced to Hawaii, antimalarial microbes may have spread through habitats shared by white-eyes and honeycreepers to colonize amakihi microbiomes, imparting their benefits to their new hosts. It is also possible that amakihi have developed unique strains of protective microbes to overcome disease. Elucidating a relationship between the microbiome and malaria susceptibility in Hawaiian honeycreepers is an essential first step in assessing the feasibility of mitigating the honeycreeper extinction crisis through the development of antimalarial probiotics. Therefore, for my thesis research, I set out to determine if variation in colloquial microbiome composition correlates with malaria infection, and identify which, if any, bacterial taxa are associated with malaria infection survival in honeycreepers and white-eyes. To address these objectives, I had 3 research questions. My first question: does colloquial microbiome diversity co-vary with malaria infection? As I stated earlier, multiple studies have established that the vertebrate gut microbiome may modulate malaria infection response, but other studies have indicated that active malaria infection can reversibly reduce microbial diversity in mouse guts. While gut microbiomes have not been explicitly examined in relation to malaria in birds, it is reasonable to suggest that this connection may extend to avian host parasite interactions. Thus, I expected to see lower microbial diversity in the cloacas of infected amakihi than in uninfected amakihi, especially those with high levels of circulating parasites. Because white-eyes do not suffer malaria morbidity, I expected to see less significant difference in colloquial microbiome diversity between infected and uninfected white-eyes. My second question: are specific bacterial taxa within the amakihi colloquial microbiome associated with malaria survivors? If specific microbes, such as those that stimulate alpha-gal antibody production, like Escherichia species, or nitric oxide production, like Lactobacillus species, are modulating malaria severity in amakihi, as was found to be the case in mice, I expected they would be more abundant in birds that survive malaria. There is also the potential that amakihi have developed novel strains of beneficial microbes that would be associated with malaria survivors. And my third question: are microbes with supported potential to modulate malaria severity in higher abundance in white-eye than in amakihi microbiomes? If malaria immunity conferring microbes, such as those of interest I've outlined earlier, are aiding in white-eye malaria resistance, it is reasonable to expect that those microbes will be in greater abundance in white-eyes than in malaria-sensitive amakihi. Hopefully by now I've convinced you that Hawaiian honeycreepers are important and worthy of conservation attention, and that there's a lot of conservation potential that could be unlocked by understanding the role of the gut microbiome in malaria susceptibility in honeycreepers. Next, I'll explain the methods I used to answer my 3 research questions aimed at unraveling the gut microbiome's role in this system. To characterize gut microbiomes, I collected colloquial swabs, which are like anal swabs. And to characterize malaria infection, I collected blood samples from 174 amakihi and 171 white-eyes. Of the various introduced bird species in Hawaii, I chose to include white-eyes as my malaria-resistant control because it is the most ecologically similar to amakihi. Variations in things like diet and habitat structure can significantly influence gut microbiome composition. But amakihi and white-eyes have a similar generalist diet consisting of nectar, insects, and fruit, and their habitats overlap completely. I also caught quite a few sassy cardinals during my efforts, which had to be given something to chew on while you handled them. Otherwise, they'd settle for chewing on your fingers, and those beaks have impressive bite strength. The swabs came in handy for this purpose as well. Birds were captured passively in mist nets and sampled from 16 sites on the island of Hawaii between February to July in 2019 and 2020 by a USGS banding crew and myself. These 16 sites were organized into 6 regions, the approximate locations for which are shown on this map. These sites varied in elevation, annual temperatures and rainfall, and forest structure, as well as malaria prevalence. I grouped these sites into larger regions based on shared geographic location to help separate out some of the environmental effects from disease effects on gut microbiome composition. Only 1 sample was retained for analysis in instances where a bird was captured and sampled more than once. Honeycreepers were rewarded for their contribution to science with a sip of nectar solution, which this little amakihi is enjoying in this picture. To characterize malaria infection, Pyrrholictum parasite load was evaluated via qPCR for each blood sample by a collaborator at Northern Arizona University. qPCR is a method for quantifying how many copies of a specific segment of DNA is present in a sample. And by targeting Pyrrholictum DNA, we can estimate the number of parasites in the blood. From the results of the qPCR, I determined the relative infection intensity among my sampled population. This was a value between 0 and 1 that represents the parasite load within each bird's bloodstream compared to the rest of the birds sampled. I then used this value to assign an infection status for each bird beyond just infected versus uninfected. Because I was looking for potentially beneficial microbes associated with malaria survival, I needed to elucidate which birds were likely capable of recovering from infection. As I briefly mentioned earlier, white-eyes are able to clear malarial parasites from their bloodstream, but amakihi remain chronically infected with low levels of parasites in their blood after exposure. Therefore, different infection statuses were assigned to the 2 host species. Uninfected amakihi in my study were most likely naive to malaria exposure. Amakihi with low-intensity infections were assumed to be those that survived malaria prior to capture but are now chronically infected, and amakihi with high-intensity infections were recently exposed individuals with active infections. In contrast, uninfected white-eyes in my study could comprise individuals either naive to malaria exposure or survivors of an infection cleared prior to capture, while infected white-eyes were likely recently exposed individuals capable of recovering. Further, studies have shown that gut microbiomes exhibit reduced diversity and microbial imbalances during the active phase of malaria, or the time when parasite loads are the highest. However, diversity and imbalances can return to baseline upon recovery. This could change how I interpret my gut microbiome data, making it even more important to classify infection status beyond just infected and uninfected. In this set of examples, the birds with the low-level infection intensity may have recovered their normal microbial communities comparable to those of naive birds, whereas the birds with active infections may still be experiencing imbalances. To identify which microbes were present in my gut microbiome samples, I extracted DNA from the colloquial swabs and used a technique called 16S rRNA gene sequencing. This is a technique that amplifies a specific region of DNA that is found in all the microbes of the gut, but is variable enough for us to be able to identify which microbes are present and in what abundances. It is not always possible to identify microbes down to the species level, so this technique classifies them into amplicon sequence variants, ASVs, which are groups of microbes whose 16S rRNA genes are identical down to the nucleotide level. I then used these taxonomic units, the ASVs, to characterize the composition of the microbes by assessing things like microbial diversity and the abundance of each ASV across samples. Now that you're familiar with the basic design that I used to begin to untangle the role of the gut microbiome in malaria susceptibility in honeycreepers, on to the fun part, the results. Of the 174 amakihi and 171 white-eyes in my study, malaria infection was detected in 49 amakihi and 39 white-eyes. Of the infected amakihi, 9 had high-intensity infections indicative of the active phase of malaria, and the other 40 were classified as malaria survivors with chronic low-level infections. To address my first question, whether gut microbiome diversity covaries with malaria infection, I measured microbial alpha diversity using Shannon diversity indices, which account for within-sample variation in ASV richness and evenness of abundance, and beta diversity using Bray-Curtis dissimilarity, which measured the differences in ASV abundance and richness between each pair of samples. I then assessed differences in Shannon diversity indices between groups using a negative binomial generalized linear model, NBGLM. This model was able to account for the overdispersion or high level of variability exhibited by the Shannon diversity index values. Because many environmental factors have been shown to be influential in shaping gut microbiomes, I was not surprised to find that microbial alpha diversity varied significantly between the different sampling regions of Hawaii. This box plot displays my selected measure of alpha diversity, the Shannon diversity index on the y-axis, and the sampling region on the x-axis. The boxes indicate the interquartile range, and the lines through them indicate the median diversity index for each population. My sites ranged from 50 to 1,700 meters in elevation and from heavily anthropogenically disturbed to nearly pristine native forest. Perhaps predictably, the highest elevation and most pristine site, Mauna Kea, had the highest microbial diversity, while the lowest elevation and most anthropogenically altered region, Puna, had the lowest. However, you can see that there were precious few samples collected in the Puna region, which likely skews the data. Low capture rate in Puna is due to a number of factors, including the fact that malaria prevalence in this region is nearly 100%. What was a little surprising was that gut microbiome alpha diversity does not differ between the two host species, nor does it vary in relation to malaria infection status for either species. This box plot again has gut microbe Shannon diversity values on the y-axis, but with infection status on the x, and samples separated by host species. You can see there are no significant differences between any of these groups. The similarity in diversity between amakihi and white-eye microbiomes may be partially explained by the similarities in their diets, and other studies have found locality to be more influential than host species in predicting gut microbiota. And while studies have found active malaria can reduce microbial diversity, they've also shown that diversity can return to baseline upon recovery. Because few of my samples showed signs of active infection, perhaps it makes sense we did not see large differences between infection statuses. However, I did not find alpha diversity covaries with relative infection intensity either. In the scatter plot, I have Shannon diversity on the y-axis and relative infection intensity values on the x. Note that the x-axis is on a log scale. The lines are based on a linear regression, and the shaded bands represent the 95% confidence interval. Neither amakihi nor white-eye microbiome alpha diversity show a significant linear relationship with malaria infection intensity. In contrast to alpha diversity, beta diversity, so the gut microbe diversity among birds, does vary between host species, in addition to varying among regions. I analyzed beta diversity with a permutational multivariate analysis of variance model, PermaNova. Briefly, this model is used to compare within group to between group variance. While sampling region only accounted for about 8% of the observed variance in beta diversity, you can see in this principal coordinate analysis that samples appeared to be loosely clustering by region, as indicated by point color, and by host species, which is indicated by point shape, and accounted for about 2% of the variance. But once again, beta diversity did not covary with infection status or intensity for either species. When I color the same principal coordinate analysis by infection status, no discernible pattern becomes clear. In sum, microbiome diversity seems to be primarily influenced by environmental factors in the various regions of Hawaii, with some differences between amakihi and white-eyes. It does not appear that malaria infection has an effect on colloquial microbiome diversity. More interesting than the gut microbiome diversity results are the patterns that I detected in the differential abundance of certain ASVs between birds of variable susceptibility to malaria mortality. For both Amakihi and White-eye microbiomes, the most abundant phyla were Proteobacteria, Firmicutes, and Actinobacteria. Differences in gut microbiome composition at the phylum level were not statistically analyzed, but using this relative abundance bar plot, we can visually see some patterns, such as perhaps some variation by region. Puna looks shockingly different with its overwhelming abundance of Proteobacteria, but again, this region had so few samples, this needs to be taken with a grain of salt. In both species, infected birds had slightly higher Proteobacteria and slightly fewer Firmicutes and Actinobacteria relative abundances than uninfected birds, which is congruent with gut microbiota imbalances seen in mammals with malaria. Proteobacteria are frequently among the most abundant bacteria in bird microbiomes, and their higher abundance in infected birds may be due to opportunistic expansion in compromised hosts. This plot shows the relative abundances of the top 10 most abundant genera in the gut microbiome samples, separated by host species and infection status. There are a lot of colors in this plot, but the take-home point here is that the top 3 genera in the malaria-resistant white-eye microbiomes, Lactobacillus, Pseudomonas, and Escherichia, are all genera that have been identified in previous studies as potentially having anti-malarial activity, whereas only one, Pseudomonas, was identified in the top genera for Amakihi. Granted, Pseudomonas was the most dominant genera in Amakihi microbiomes by far, accounting for 32% of the total relative abundance. This difference in abundance at the genus level is interesting, but again, they were not statistically tested. To answer my second and third research questions, assessing if there are specific microbes associated with malaria survival in Amakihi or white-eye gut microbiomes, I had to look a little closer, down to the ASV level. To do this, I used a walled test to look for differential ASV abundances between groups of varying malaria susceptibility. For question 2, whether specific microbes are associated with Amakihi that survive malaria, I ran the differential abundance analysis between chronically infected Amakihi, who are malaria survivors, and uninfected malaria-naive Amakihi. I discovered 14 ASVs that were significantly more abundant in the survivors. Each point of this plot represents one of these 14 ASVs, with the genera it belongs to on the X and the phylum indicated by the color. The y-axis is the log base 2-fold increase in abundance between chronically infected and uninfected Amakihi. None of these differentially abundant microbes were Escherichia or Lactobacillus species, suggesting these microbes may not be playing a large role in increased Amakihi survival. While Pseudomonas species do not appear in this analysis either, I did find by linear model that the abundance of 2 Pseudomonas species within Amakihi microbiomes were positively associated with disease pressure at sampling sites. The scatter plot has malaria prevalence measured as the percent of native birds positive for malarial parasites at each site on the y-axis and normalized ASV abundance counts on the x-axis. Of all the bacteria in the colloquial microbiomes of Amakihi, these Pseudomonas species were 2 of the only 3 that were significantly more abundant in areas with high malaria prevalence. The third was an ASV that could not be identified beyond the class level, an alpha proteobacteria, but its association with malaria was weak, if statistically significant. Selective disease pressure can favor rapid adaptation of immune strategies. And recall, there's evidence from mosquito studies that Pseudomonas can block parasite development. It is possible that harboring a greater abundance of these Pseudomonas species is advantageous in surviving infection. Thus, in areas with heavy disease pressure, Amakihi are evolving more Pseudomonas-dominated microbiomes. To answer question 3, are microbes that have shown anti-malarial potential in greater abundance in white-eye microbiomes than in Amakihi microbiomes, I ran a differential abundance analysis between uninfected white-eyes and uninfected malaria-naive Amakihi to remove the potential confounding factor that active infection is causing microbial imbalances. I found 25 ASVs were positively associated with white-eyes. In this differential abundance plot, we can see that 2 bacterial genera of interest to my study each have at least 1 ASV showing greater abundance in white-eyes. 4 Lactobacillus species and an Escherichia species, as well as 2 ASVs in the same family as Escherichia and Enterobacteriaceae that could not be identified to the genus level, were significantly more abundant in uninfected white-eyes than in Amakihi. I also compared uninfected white-eyes to chronically infected Amakihi to see if microbial abundances of Amakihi that had survived malaria infection were more similar to white-eyes than the pool of malaria-naive Amakihi. This did not appear to be the case, however, as I found 48 ASVs that were positively associated with white-eyes, nearly double that of the uninfected comparison. Of these, 5 were lactobacillus, 1 Escherichia, as well as another unidentified Enterobacteriaceae, and even 1 Pseudomonas species. The ability of these microbes to prime host immunity may be working synergistically with white-eye genetics and behaviors to protect them from malaria mortality, but do not seem to be the strategy that surviving Amakihi have adapted. My study is among the first to assess correlates between malaria infection and microbiome composition in avian hosts sampled in a natural system. To summarize my key findings, I found no difference in colloquial microbiome alpha diversity between Hawaiʻi Amakihi and warbling white-eyes, and only a small yet significant difference in beta diversity. My results suggest microbiome diversity is predominantly shaped by environmental factors in bird habitats, which aligns with several other avian microbiome studies looking at birds with similar diets. While microbiome diversity did not covary with malaria infection, I identified a total of 71 bacterial ASVs that were more abundant in groups likely to possess some immunity to malaria mortality. I have shown that the malaria-resistant white-eye harbors microbes with established connections to malaria immunity in greater abundance than malaria-sensitive Amakihi. Escherichia and Lactobacillus species were among the top genera in white-eye microbiomes, accounting for 11% and 19% ASV relative abundance respectively, but both were much less abundant, only 1 to 2% in Amakihi. Further, an Escherichia species, as well as 3 ASVs in the same family, Enterobacteriaceae, and 5 Lactobacillus species were significantly more abundant in white-eyes than in Amakihi. Of course, there may be other microbes that can confer malaria resistance, as there were dozens of white-eye associated ASVs that I have not highlighted today. But both of these genera of bacteria have ample evidence from mammalian systems of being capable of reducing malaria transmission and severity by priming host immune systems to either recognize parasites through the stimulation of cross-reactive alpha-gal antibody production or suppress parasites, potentially through the stimulation of nitric oxide production. The fact that they did not appear in my comparison between survivor and naive Amakihi suggests they have not spread from white-eye microbiomes to native birds, and that potentially a different immune strategy has evolved in Amakihi populations with low mortality rates. There were 14 ASVs associated with malaria survival in Amakihi microbiomes, though none with previously established connections to malaria. However, Pseudomonas was the top genera in Amakihi by far, accounting for 32% overall relative ASV abundance. What's more, chronically infected survivors had nearly double the relative abundance, 46%, compared to naive birds only 27%. This difference is just an observation without statistical backing, but it does paint an interesting picture. Further, there were 2 ASVs in the Pseudomonas genus that were significantly positively associated with malaria prevalence in habitats, meaning they increased in abundance as selective disease pressure increased. Both in vivo experiments in the midguts of mosquitoes and in vitro experiments in co-cultures of malaria parasites and bacteria have demonstrated that Pseudomonas species can block parasite development. It may be it blocks malaria development by outcompeting parasites for resources, in which case it may not provide protection in host microbiomes. In mosquitoes, malaria parasites invade the gastrointestinal tract, but in hosts, they invade through the skin into the bloodstream and would never be exposed to resource competition from gut microbiota. However, if Pseudomonas secretions are able to enter host bloodstreams and act directly on parasites to suppress their growth, or if they're able to stimulate host immune systems in some way, they may be providing benefits, and having an overabundance of Pseudomonas species in your gut could prevent malaria infections. I'd like to conclude by highlighting some of the future research that could build upon my work and the conservation implications of my findings. My foundational study has uncovered candidate microbes that warrant further investigation to elucidate the benefit of supplementing malaria-susceptible Hawaiian honeycreepers with immune-modulating bacteria. That I was able to find significant associations between colloquial microbiome composition and malaria survival in my correlational study suggests that lab experiments may be highly revealing and quite fruitful for conservation. In addition to allowing us to test if supplementation of any of my candidate microbes can lower malaria mortality rates in birds, controlled laboratory experiments would allow us to sample the microbiomes of birds that succumb to malaria mortality, which was not possible for my study. Because I did not have fatality samples, I used comparisons between chronically infected survivors and uninfected malaria-naive birds to identify microbes that may be augmenting malaria immunity. My pool of naive amakihi likely consists of a mixture of birds that would either survive or die upon malaria exposure, which may have weakened my ability to detect if beneficial microbes are in greater abundance in survivors. In future lab experiments, it may also be feasible to sample other regions of the gut. Microbes that have been found to be influential in malaria immunity have mainly been members of mammalian intestinal communities. However, I used cloacal microbiome samples because they are substantially less invasive to collect and were logistically practical. Cloacal microbiomes are not perfect representatives of the microbial community in other regions of the gut, as we can see from these alpha diversity box plots and relative abundance bar plots from a study of ostrich gut microbiomes. Despite the added challenge, sampling the ileum, cecum, or colon in laboratory experiments may reveal even stronger associations between commensal microbes and infection response. The number of avian microbiome studies is exponentially growing, and as more bacterial genetic data is accumulated, future studies like mine will be able to conduct more comprehensive gut microbiome characterizations and analyses. Currently, there's a lack of characterized avian microbes in taxonomic databases, which poses a prevailing limitation to contemporary gut microbiome studies. A notable number of ASVs in my study could not be taxonomically assigned, even to the phylum level, but my data could be revisited once a greater number of microbes are described. To my knowledge, the use of probiotics to mitigate disease has not yet been attempted for wildlife populations. However, the prospect is gaining scientific attention and traction among wildlife managers. Orally delivered probiotics have been effective in mitigating disease experimentally with mice and poultry models and are used extensively in livestock operations. Probiotic supplements may provide a way to avoid a labor-intensive malaria control strategy, such as capturing and vaccinating entire honeycreeper populations. Except for very small populations with limited ranges, vaccinating enough birds to prevent extinctions may not even be possible. Targeted probiotics that could be administered passively in food supplementations to endangered honeycreepers in the wild and in captive breeding programs may be an effective and practical approach to bolstering malaria immunity in the face of increasing disease pressure in Hawaii. Lactobacillus species in particular show promise, as they're frequently used as probiotics in other systems and are less likely than Escherichia species to become opportunistic pathogens. Pseudomonas species may also be valuable candidates for probiotics, as they appear to be naturally abundant in amakihi microbiomes, especially in areas with high malaria prevalence, and could represent a unique immune strategy evolving in amakihi. The beautiful, diverse, and ecologically critical Hawaiian honeycreepers are in the midst of an extinction crisis, and investing in research and development of innovative conservation strategies needs to be of top priority. I believe my study has elucidated a potentially effective and practical method of augmenting immunity in malaria-sensitive species on the brink of extinction, and I'm excited to see what future probiotic studies can yield. This has been a huge project, which I could not have completed without the help and support of numerous organizations and individuals. My research was funded by the Smithsonian Conservation Biology Institute, the Yoshimoto Foundation Charitable Trust, the American Ornithological Society, and the National Science Foundation. Throughout my research, I've had the opportunity to work with so many amazing scientists. I can't even list them all here. I want to mahalo my thesis committee, Pat, Christina, Eben, and John, for your guidance and feedback. Pat, I am so grateful for the opportunity to have been your student and a member of your lab. I have grown so much as a scientist during this time, and you have been an absolutely wonderful advisor. Eben, your expertise as I was developing research questions and analyzing data allowed me to look at deeper connections than I originally saw. I genuinely appreciate your feedback. Christina, words cannot express how thankful I am to you. You've been such an amazing role model for me and have opened so many doors for my future. It means the world to me that you saw potential in me and have set me on this path that has been so stimulating and rewarding. Thank you. To my Smithsonian Fellowship Advisor, Rob, I want to thank you for the opportunity to work with you in DC. It was such a unique opportunity for me to study at the CCG last year. I learned so much, and I truly hope we work together again in the future. And Nancy, who helped me get set up at the CCG, I couldn't have done it without you. Thank you so much, and for sequencing my samples. I want to thank Carly from the CCG as well, who was so ready to answer any microbiome questions I might have while I was there. To my Smithsonian mentor, Aylin, you have been an amazing mentor and advocate for me, being available at the drop of the hat to help me and guide me, for providing me with ample opportunities and celebrating my successes. You have been absolutely wonderful. Thank you for all that you've done. I also want to mahalo my collaborators at Northern Arizona University, Jeff and Katie, who processed all the blood samples for my projects, as well as the USGS Avian Malaria Genomics Research Field Crew led by Liz, who helped me develop my bird banding skills and taught me how not to lose a fingernail to cardinals. A big mahalo to Jared at the UH Genomics Lab for helping me get set up there and saving my skin when I ran out of enzymes while processing a huge batch of samples. And Anne, thank you for being the greatest Lohe Lab ambassador anyone could ask for, ready with a big smile and helpful attitude. I think we can all thank you for the beautiful bird pictures I've used in my PowerPoint. It would be amiss for me to forget all my wonderful classmates in the 2019 TCBES cohort, whose support and friendship got me through this degree, especially that first semester when none of us knew what we had gotten ourselves into. Lastly, I want to thank my beautiful, wonderful, supportive, amazing friends and family for always being there for me. The mental health walks with Robbie, the chats with my sisters, Mary, Carrie, and Audrey, and my Madison ladies, Amanda and Marissa, and the emergency ice cream parties with Ben were the things that helped me stay sane as my perfectionism nearly drove me to insanity. And I cannot tell you how much I appreciate each and every one of you. I hope I have made my parents, John and Kate, proud. They always told me that I could do anything I set my mind to, and I believed them. It is because of their love and support I was able to accomplish what I have. There are certainly others I just do not have time to name, but thank you to anyone who has helped me get to where I am. I truly appreciate you. And I want to thank all of you for listening. And with that, I will take any questions.